So our project was called Microcolors. Sorry, my project partner Boram can't be with us. She's actually on a flight at the moment. But yeah, our project was called Microcolors. We wanted to see if colour-fast fabric dyes could be created from pigmented microorganisms. We hope to find a solution to the current harmful dyeing and printing methods used within the industry. So we teamed up with 2 microbiology students who helped us with the science side of things. Our current dyeing process in the textile industry is linear. We take a raw material, we make products using environmentally harmful processes, and then dispose of the product at the end of the life. But Microcolors focuses on creating a circular material cycle to reduce environmental impact within the industry. So our aims were to reduce chemical impact, to reduce energy and water use, and explore cleaner and better technologies. So our first job was to find, extract and grow the microorganisms. Pigmented microorganisms can be found on pretty much anything in the world, but some are quite dangerous, so don't try and grow any at home. So these are the organisms we used. R.rubra was the pink yeast, which the lab provided. The blue one is L.casei, and it's a bacteria, and M.luteus was the yellow one and from soil samples. The blue one, sorry, was from yogurt. So it is one type of microbe from a sample. We were in the lab a lot and tried many different experiments. They all grow at different rates, temperatures, and on different agars. So it's quite complicated to keep track of all the experiments all happening at the same time. But it is also important to know that once you have extracted the pigmented microorganism you want, you can keep growing it, so you don't have to keep sourcing it again and again, unless you can't let it die. So now we have the organisms. We wanted to see if they would dye the textiles and if we could design with them. So first we wanted to see if we could get full coverage of dye on the fabric. It was unsuccessful because we didn't have enough organisms in the solution, and it was too diluted. So in our next experiment, we wanted to extract pigment from both liquid and powder form. It did work, however, in such small amounts, it wouldn't have worked onto fabric. So then we realized we would need to use the microorganisms without diluting it if we wanted strong color. So we tried massaging the organisms directly onto the fabric surface. This worked well, and we got even coverage of dye and bright color. This was the result after they were sterilized. All samples must be sterilised, so they're safe to touch. We tried some sun tests and tried them in the washing machine, and the colour didn't fade, so we were really happy that we found that. Next, we wanted to try different dye methods to see if we could design with the microorganisms, and here are some of our results. We wanted to know if the organisms would grow a design onto the fabric surface. Here you can see I'm pipetting the organisms onto the plates with the fabric and the yarn inside. So this is what they look like in the Petri dishes after the organisms had grown onto the textiles. We tried folding the fabric, twisting it, scrunching it. The only problem with this method is we were limited to the size of the plates, therefore couldn't make large samples. Here is the results. It does look a bit like tie-dye, but it is kind of interesting to see how the organisms grew straight onto it, but you couldn't get full coverage of dye. Next, we tried folding, twisting, scrunching, clamping, origami, and shibori techniques for larger samples, using the organisms straight from the Petri dishes. Here are some of our results. Some worked better than others, but we think the shibori was quite good. So 1 litre of dye, 1 litre of water can dye 4.6 metres squared of fabric. No chemicals, mordants or textile treatments are used within our dyeing process. And microbial dyes have huge potential and could be the solution. If our process was scaled up to an industrial context, there is the possibility of extracting a large range of colours from locally sourced organisms. Thanks so much for listening. We're really grateful that we've been able to share our work with you. If you've got any questions, let me know.